Okay, so there are 50 questions are there. First, I will explain you 15 questions. Then I'll give the time to ask any kind of a doubts. Once again, we will continue. Okay, so now listen. Okay, see, most of the people really did well. Okay, but some people, did not performed well. The people who did not performed well, the people who scored below 60 has to work very hard. And people who are in the 60 to 65 also, try your best to get more marks. Okay, and whatever the questions that we got now, the same kind of questions only will appear in the JL and DL level. So I want every one of you to understand the concept and understand the given question, and I want you to analyze it very slowly. Okay, and even though the people who scored above 80 also, no one has not answered some of the most difficult and logical questions. Now, let me tell you how to answer all those questions. Okay, now let's continue. The question number 1 says that which of the following pair is incorrectly matched? So friends, you know very well we are having a two type of a bases. We can call one is purines and one is the pyrimidines. And you know very well purines are the adenine and guanine and pyrimidines are the cytosine and uracil. Option A is correct. Option B is also correct. Nucleoside means what, friends? Sugar plus base together we can call it as the nucleoside. So which means there is no phosphate group is present. So when the base adenine combine with the sugar, we can call it adenosine. When the base thymine combine with the sugar, we can call it thymidine. So obviously this is also absolutely true. Then the option 4 is what? DNA is a basic molecule. This is wrong, right? DNA is a acidic molecule. Histones are the positively charged. That's why they will easily associate. Anyway, remember here DNA is the acidic in nature, not to get confused with that point. Hence the correct answer is option D. Next one, let me explain you 15 questions. After that, I'll solve your doubts. Next question, friends. The question asked, the given flow chart represents the flow of genetic information between biomolecule. Identify the A, B, C, D and select the correct option. You know very well, A means what, friends? The DNA undergoing the process of replication. It's very simple. B is what? DNA giving rise to the mRNA, which we can call transcription. And if you look into the C, you can see very clearly the mRNA is going to give rise to the DNA, right? So which is obviously reverse transcription. And if you look into the mRNA, mRNA is going to give rise to the protein. Then what we call it that as, we will be calling that as the translation. So replication, transcription, reverse transcription, and translation. Now you only see where it is present. Of course, option C will be the correct. Replication, transcription, reverse transcription, and translation. Okay. So the only answer is C. C is correct. Histone proteins. Every one of you know very well. Why, friends, histone proteins, suppose this is the DNA molecule. Why the histone proteins wrap around the DNA? What is the reason? Because histone proteins are basic and positively charged. And I want every student has to remember for the exam point of view, histones are basic in nature, right? So they majorly contain amino acids such as the lysine amino acid as well as the arginine amino acid. Not to get confused with that, because these two are the basic amino acid. Now, if you look into the next question, question number 4, refer the given sequence of the steps and select the correct option. See, friends, now you only try to look into this. DNA is going to give rise to hnRNA, and some students given this also wrong statement. Friends, first of all, what is the difference between the hnRNA and mRNA? Very simple, friends. hnRNA nothing but inactive RNA. Widely called it as the immature RNA. And friends, the point is very, very clear. When the immature RNA can be converted into mature RNA, when immature RNA can be converted into mature RNA, once the processing occur, correct na? So the processing such as 5' capping, splicing, and 3' polyadenylation. So, but whether it is a hnRNA or mRNA, what is the first point? It is RNA. So synthesizing of RNA from the DNA is what? Undoubtedly transcription. Now, inactive mRNA, we are converting into active mRNA. How can we see that? How can we convert the inactive mRNA into active mRNA? By doing the process of splicing, which is one of the modification. Then mRNA is going to give rise to what? Proteins, which is translation. Hence, the correct answer is C. Simple point. Now, question number 5. Everyone pay attention. Chemically, RNA is dash reactive and dash stable. I have already told you, friends. DNA is the highly stable molecule. Whichever is highly stable, what you have to remember, it is more reactive. What about the RNA? RNA stability is very low. Low stability, friends. As a result, what you have to understand, it will be more reactive, not to get confused. In terms of the reactivity, not to get confused here, stability is entirely different. Reactivity is entirely different. So when I'm talking about the stability, I'm talking about the lifespan. Okay. So when compared to the DNA, RNA having the very less lifespan. You remember, I told you that mRNA can withstand up to hours. tRNA can withstand up to only minutes. Only rRNA can withstand up to generations. But the point is what? In terms of the stability, they are extremely more stable. No need to get confused with this particular point. Which is more stable? DNA is the more stable. RNA is the less stable. Whichever having the less stability, what you people has to understand, maybe reactive wise, it is very faster. But lifespan wise, it is very low. Not to get confused with this point. So what we can say that RNA is more reactive, but it is less stable. And one more point, everyone remember, when I use the word less stable, also remember less lifespan. That also you have to remember. And also you have to remember degrade very fast. So that's why DNA never degrade very fast when compared to the RNA molecule. Not to get confused with that. So which is a more reactive and less stable. Easy to remember. Option C is the correct one. Now, last time also we have studied this. What does the given diagram represents? You all people studied many number of times. What is nucleosome represents? Nucleosome represents DNA plus histone proteins, including the linker also. You know, right? This is the histone one, which is acting as a linker protein. DNA plus histone protein together. Because in the diagram, you can see DNA also. You can see histone proteins also. That's why we can call it nucleosome. Spliceosome means what? The proteins of different splicing proteins, which consist of RNA plus protein. Correct, na? In the nucleosome, you can't see any RNA, only DNA and histones. Histone complex is not the answer. Both A and B is not the answer. The correct answer is option A, nucleosome, because you can see only DNA and histones you can see, right? Now, this question also some students has given the wrong answer. Friends, it is very easy, right? Every one of you focus on the question number 7. Which of the following shows the correct positions of the phosphate, sugar, and base molecules in the line diagram representing the structure of DNA? How many number of times I have told you? Friends, phosphate to sugar is the backbone. Every student remember, imagine friends, this is one nucleotide having sugar, base, and phosphate, is it? And this is the another nucleotide, is it? It is also having sugar, then followed by the base, and then followed by the phosphate group. So what is the point I want you to understand here? A very important and interesting point. So what is this starting from 5', which is the phosphate group? Imagine friends, this is only last nucleotide. Here what group you can see? Here you can see sugar, OH group. So always remember, so phosphate to sugar, phosphate to sugar, phosphate to sugar. This is called as backbone of the DNA. To this, you know, right? Always to the sugar, base will be attached. To the phosphate, never ever be base will be attached. Every student know that. Only to the sugar, you can see, right? Only to the sugar base is getting attached, undoubtedly. So what will be the right answer here? The option 1 is the correct answer. Option B, they says that it is starting with base. That is also wrong. Option C says that sugar is attaching to the base. That is also wrong. Option C says that it will start with the sugar, then followed by the phosphate. They are showing that phosphate is associated with the base. That is wrong. Always remember, sugar always associated with the base, and the backbone start from phosphate to sugar. Hence, the option 1 is the correct. Very simple, little logical question. And you know very well, the question says that what in the question number 8, in a DNA molecule, the phosphate group is attached to the carbon dash of the sugar residue of its own nucleotide and carbon of the dash sugar residue to the next nucleotide by dash bonds. Friends, you know, right? Say, just now I explained you. This is sugar. This is what? A base. And this is what? Phosphate group. What is the first question they asked? In DNA molecule, the phosphate group is attached to the carbon of dash of the sugar. First carbon attaches with the sugar. Fifth carbon attaches with the phosphate. So it is the fifth carbon. And carbon of the sugar residue to the next nucleotide. This is what group? OH group. OH group will go and associate with the phosphate group of the next sugar. Then this is at the OH group is present at which carbon? Third carbon. What do you call this bond? Phosphodiester bond. So 3 prime, then phosphodiester bond. Hence, what is the correct answer? 5 prime to 3 prime phosphodiester bond. Very simple, straightforward, and direct question. This also some people has given the wrong. And you only see here. What does A and B represents in the given representation? How many number of times we all people studied, friends? When sugar and base, only if these two are present, what do we call the side, nucleoside? When sugar and base associated with the phosphoric acid, what do we call it as, friends? We will be calling it as the nucleotide. Correct, na? So A will become nucleoside and B will become the nucleotide. Is it or not? Hence, this will be the correct answer. Now, question number 10. This also some people given mistake. I'm not understanding. Actually, 4 to 5 people has given this question also wrong. Think it correctly. The question says that the given flowchart shows central dogma. They are asking, remember everyone, they are asking, they have given DNA, they have given RNA, and what is the question they asked? Last time also I have explained in the last test also. They asked is about enzymes. How many number of times I told to all of you? Once again, I'm reminding you. The moment you heard the name of a replication, replication means what? We are synthesizing DNA. So who will synthesize? DNA polymerase will synthesize. It is getting the signal from where? DNA only, right? So you can call DNA dependent DNA polymerase. Simple point. This is the replication. When we are doing the transcription, what we are synthesizing? RNA. So who will synthesize it? RNA polymerase will synthesize it. But signal coming from what? Only DNA. So DNA dependent RNA polymerase. When you look into the reverse transcription, tell me what we are synthesizing. We are synthesizing DNA only. So the enzyme is DNA polymerase only. But what is the enzyme? The enzyme is reverse, which is RNA dependent DNA polymerase. Not to get confused with this particular point. Is it? Very straightforward question. So A is what? DNA dependent DNA polymerase. B will become what? DNA dependent RNA polymerase involved in the transcription. Reverse transcription, RNA dependent DNA polymerase. Hence, this will be the correct answer. Now, question number 11, which is a very straightforward, no need to explain here. The question asked is, I told you, right, you people has to remember the name of the scientist compulsory. Match column 1 with column 2 and select the option with the quotes given below. You all people commonly know it. So Frederick Mischer. You know, no friends, Frederick Mischer is the first scientist, I will tell you in the biomolecules, who isolated, all the students listen my words correctly. Frederick Mischer is the scientist who isolated a substance, from the nucleus of pus cell. From which cell? From the nucleus of the pus cell, he isolated a substance and he has given the name nuclein. So that's what it is. Okay, Frederick Mischer, nuclein. You all people commonly know about it, and everyone know very commonly. Griffith performed the experiment by using a Streptococcus pneumoniae, is it? And Hershey and Chase on the bacteriophage. And you all people commonly know that Watson and Crick will be DNA double helix. Rosalind Wilkins and James Franklin study the X-ray diffraction studies. Hence, the correct answer will be the option A. Very simple, straightforward question. What is the question number 12 says? Watson and Crick proposed a DNA double helix molecule and won the Nobel Prize. The question says that their model of the DNA was based on what? You know, no, right? In the first class of molecular biology, I told you very clearly. They have proposed the double helical structure based on the X-ray diffraction. And also what we understood, based on what rule they proposed. They proposed a rule that number of purines are always equal to the number of pyrimidines in the DNA. Who proposed this? Chargaffs. So X-ray diffraction studies done by the Wilkins and Franklin. This is very important to understand the DNA structure, and Chargaff's rule. These two are major. And anyway, you can eliminate the Griffith transformation. Some people has chosen the Michelson and Stahl also. How is that possible? Michelson and Stahl function is what? To prove what? We studied, right? To prove the semi-conservative model of the DNA replication only, Michelson and Stahl experiment is used. But that is not the question asked. Question asked is DNA double helix model is proposed based on what? Based on X-ray diffraction studies, based on the Chargaff's rule. Hence, the correct answer is option 1 and 2. This is the answer. And you know very well. They're asking question, select the incorrect statement regarding DNA replication. They're asking incorrect, friends. Every student commonly know very well both the leading strand and the lagging strand. What did I told you? Both occur in 5' to 3' only, but one move away from the replication, one move towards the replication. So option A says that leading strand is formed in 5' to 3', right? Okazaki fragments are the lagging strands formed in 5' to 3', right statement. Okazaki fragments are formed in 5' to, why this option got repeated? Anyway, this is also right. Option D says that DNA polymerase catalyzes polymerization in 3' to 5' direction. Wrong, right? DNA polymerase always synthesize the daughter strand only in 5' to 3' direction. Hence, the incorrect option is this, which is the D. Now, select the correct match with related to its enzyme function. DNA polymerase function is what? Polymerization or synthesizing the new strand. Absolutely true statement. There's no doubt about it. DNA polymerase synthesize the synthesis. Why it is getting repeated? I'm not understanding. Yeah, of course, synthesis of the new strand. Ligase. Ligase is the function is what? Joining the phosphodiester bond. Nothing but joining one fragment with the another fragment. Of course, that is also true. Hence, what will be the answer? Answer will be the D. I think here helicase it is. Helicase function is unwinding the DNA. DNA polymerase synthesizing the new strand. Ligase will join the strand. Undoubtedly, the correct answer is all are correct. And surprisingly, I'm not understanding. I have explained you many times. 2 to 3 people has given this question also very wrong. I'm not understanding. Which of the following figure correctly represents the replication fork formed during DNA replication? How many number of times I told you? Friends, one strand will move always towards the replication fork. Suppose this is the replication fork. What about the another strand? It is coming away from the replication fork. But what is the common point? Both will be synthesized in the 5' to 3' direction only. Correct, na? Not to get confused. Why you are getting confused, I am not getting here. You see here, one is moving towards the replication fork. So 5 prime, 3 prime, right? And what will be the opposite one? This will become 5 prime, this will become 3 prime. So 5 prime to 3 prime. Is it or not? So here both are moving in the same direction, right? In the third graph. So it is wrong. Here, this is a 5 prime, and here also both are moving in the same direction. This is also wrong. Here it is showing a 3 prime to 5 prime. That is also wrong statement. So only the option D will be the correct. One is moving in 5 prime to 3 prime and another also moving in 5 prime to 3 prime, but both are moving in the opposite directions. Hence the correct answer is option D. 2 to 3 people has given this question also wrong. I want you to look into that. Now see, in this diagram, you have to identify what is A, what is B, what is C, what is D. How easy to remember? You all people commonly know that, friends. With what our, what we can say that transcription will start. You all people know very well, RNA polymerase enzyme will come and associated to the region called promoter. So promoter is the starting one. So you're all able to see this arrow, right? INR, you remember, wherever the transcription is starting, initiation region of transcription. You know very well where the transcription will be end, friends, at a point called termination. So the point B is called as what? Termination point. Not to get confused with that. And you all people know very well. Some students surprisingly made this also wrong. I am not understanding how. How many number of times I told you RNA has to be synthesized in which direction? 5 prime to 3 prime. Yes or no? So now what kind of strand you have to use from the DNA? You have to use the 3 prime to 5 prime. Yes or no? Whichever you use, that will become what? That will become template. So here C is in which direction? Every student. C is in 3 prime to 5 prime. Then that will become what strand? Template strand. How many times I told to all of you? Friends, template means non-coding. Then option D, the D strand will become what? Coding strand. Simple point it is. Hence, what will be the correct answer? Promoter, terminator, template, and coding. Option C will be the correct answer. Very simple, straightforward question. I don't know how this also people has made it wrong. How you made it wrong? Very easy and straightforward question, right? It is also. Right. So now you only see your A. And see, first A is very common, right? At the 5 prime end only, we will add the cap. So A will be undoubtedly methylated cap only. Is it or not? Of course, yes. Now, what will be the B? So you remember, B will be the non-coding region. Why? Because that strand also in 5 prime to 3 prime. So then that will become what? Non-coding region, not to get confused. Then in the previous slide also same thing we studied, right? C become what? Coding region. And D becomes what? Poly A tail. This also people got confused. All the time you remember, DNA having two strands, 3 prime to 5 prime, 5 prime to 3 prime. Every one of you keep the point in your mind. Whenever we are synthesizing the RNA, we always use 3 prime to 5 prime strand of the DNA as the template strand. Keep the point in your mind all the time. Whichever is template, that is non-coding. And 5 prime to 3 prime, it is not acting like a template. Correct or not? So it can act like a coding strand. So here B will be the, what we can say, non-coding region. C will be the coding region. So obviously answer will be the C. This also people made it wrong. Think it correctly. Slowly you have to analyze. C is the correct. Now you know very well the structural genes in eukaryotes possess coding and non-coding sequences. Coding sequences we can call the exons, and the non-coding sequences what we people generally call it as the introns. Hence the correct answer will be C. Is it? Of course, yes. Now you no need to think it also, right? You can easily answer this particular question. Sigma factor. Sigma factor function is what, friends? To start the process of transcription. So it is required for initiation. You know, right? Capping means what, friends? We are adding the cap at which direction of mRNA? 5' direction of the mRNA, we will add the cap. Where we add the tail we will add the 3' end. Just now I told you, right? Coding strand never act like a template. So coding strand will be always in the 5' to 3' direction. Not to get confused with this particular point. Easy to interpret, right? So it's very easy and straightforward question. And this also people has made it wrong. I'm not understanding how. See, what people are thinking, everyone think A is the RNA which is coming out. And what every student think, C is the sigma factor. You tell me how the C will become sigma factor. Friends, use your brain. Think it correctly. Suppose if C is the sigma factor, then where the RNA polymerase enzyme is present? It is present here. But what you can see in the diagram, RNA synthesis is over. RNA came out. If RNA came out, this particular RNA polymerase has to dissociate from the DNA. If it wants to terminate, what factor it requires? We studied, right? Rho factor it requires. How does it become sigma factor? This also people made it wrong. So if you see RNA polymerase here, if it is associated with the DNA, then that will become the sigma factor. This also people made it wrong. So A is RNA, B is RNA polymerase, C is the rho factor. Easy to understand this, right? This also people has given the wrong. What is there to explain? I know it's very easy, right? Every student understand my words correctly. Whenever sigma factor is present, all the students use your brain. Whenever the sigma factor is present, RNA synthesis is happening, which means what? RNA still attaches to the DNA. Everyone use your brain. Here what you can observe, here RNA is not associated with the DNA. Where you can see RNA synthesis is over. It came out. RNA synthesis is over. If RNA synthesis is over, what is the use of RNA polymerase enzyme on the DNA? It also has to come out. How will it come out? If it wants to come out, the termination has to be happen. How does the termination is depends on? The termination is actually depends on the sigma factor. Yes or no? Of course, yes. Sorry, termination is depends on the not sigma, rho factor. Initiation is depends on the sigma factor. Then obviously the C will become what? C will become the rho factor. Understand? It's direct question. Now you only tell me. Friends, you no need to think it, right? If you look into this diagram, what you are observing? You are observing that post-transcriptional processes are happening. One point is very clear. We all know very well. Post-transcriptional processes never ever takes place in the prokaryotes. You all know very well. So what is the answer you have to choose? It is a, remember everyone, transcription which is happening in the eukaryotes. Instead of the transcription, if they use the word post-transcription, that also you can use. Why? Because capping, polyadenylation, splicing, it never ever takes place in the prokaryotic organisms, which is only in the eukaryotes. But why they used the word transcription? Because they're all happening in the during the synthesis of RNA. That's why they used the word transcription. If they give the option of post-transcription, also correct. No need to get confused over there. Now question number 22. This also some people has given wrong answer. Friends, it's very easy, right? This is the coding strand of DNA. This is the non-coding strand of the DNA. How many times I am continuously telling all of you, whichever is the non-coding strand of the DNA, that is the only strand which can act as a template for the mRNA synthesis, because non-coding strand is in the 3' to 5'. So mRNA can be synthesized in which direction? 5 prime to 3 prime. So you won't use this strand because this is also same strand, same direction of mRNA. So how can we use this? You can't use this, right? So we have to use only 3 prime to 5 prime. Now you only see the opposite. So 5 prime, C pair with G, C pair, C with G, G with C, T with A with U, U, C, G, G, C, U, A, G, U, A, C, 3 prime. Now you only see where it is located. So this will be the answer. Easily you can interpret the question. Very straightforward, very, very direct question. Hence the correct answer is option 1. Hope everyone is getting correctly. Ask me if you don't understand. And this question is also very good and very, simple question. Select the correct statements regarding the process of transcription in eukaryotic organism. Option 1 says that the strand of double-stranded DNA which takes part in the transcription process is called coding strand. Every student say yes or no. Is this a true statement or false statement? So can the template strand is called as coding strand? Everyone, is it true or false statement? So everyone, obviously it is a false statement. Coding strand you never call template strand. Always remember, non-coding strand only you called it as the template strand. So option 1 is wrong option. The enzyme RNA polymerase catalyze the polymerization only in one direction, 5 prime to 3 prime, right? An unusual nucleotide 5, what we will add? Methyl guanine triphosphate. Yes or no? Added to the hnRNA. Nothing but immature RNA to make it active. Obviously, option 3 also right. Option 4, during tailing process, adenylate residues. We studied, right? Poly A. We add 200 to 300 adenines at the 3 prime end. So that is also right. Correct. Hence the correct answer is option 3. Easy it is. The fully processed hRNA is called as what? Fully processed means what, friends? Processing is over. Whichever processing is over, we can call mRNA. Correct, na? Whichever the processing is over, from the nucleus, where it enter, friends, it will enter into the cytoplasm. Why it has to enter into the cytoplasm from the nucleus? Because cytoplasm is the region where the translation will be taking place. Yes or no? Hence the correct answer will be number 1. Very simple, straightforward question. Select the incorrectly matched. This also some students, made it wrong. AUG, GUG. GUG actually going to code for valine. In some organisms, I told you, I don't know whether you remember or not. I have given you one statement. Genetic code is present everywhere, but not universal. Means it is not going to code for the same information everywhere. In most of the mRNA, AUG is the start codon. In some of the mRNA, GUG is the start codon. So both are initiation codons. That is right. Stop codons, there are 3 stop codon: UAA, UAG, UGA. This is also right. Methionine, which is the AUG, anticodons, mRNA. That is wrong, right? mRNA contains what, friends? mRNA contains codons. Who is bringing the anticodon? You know very well. Anticodons brought by the tRNA molecule. Not to get confused with that point. Right. So that's also correct. Next, the 3 codons. So correct. The wrong answer is option D. The 3 codons which result in the termination: UAA, UAG, UGA. That is also right. Now, 75% of the people have not answered this particular question. Why? Because no one studied. I told you it's your rest. See, if you are reading biochemistry or if you're reading molecular biology, it is very important. You people has to read 64 codons compulsory. That's why most of the people are unable to answer. I know, except this, you don't know anything here. Right. So that is not the correct way. You have to study the 64 codons compulsory. AUG. What does the AUG? This is the only thing you people know it. What AUG actually code for? AUG code for methionine. Any one of you very quickly tell me what does this particular U code for? All the students remember U will code for the very good. U code for what? Phenylalanine. Not to get confused with that point. GGG is for the glycine, is it? Then UCU, it is code for the serine. Then CCC, it is the one which is coding for proline. AUG is going to code for what? Methionine. Is it? Not to get confused, friends. You have to remember 64 codons compulsory. It's very important. Next, it is very easy, right? Whenever the tRNA undergo the modification, we studied that it is going to give rise to a, what I told you, clover leaf like structure. How many number of times I have explained it to all of you? Wherever the 3' hydroxyl group is present, there only who will go and associated? Amino acid will go and associate. This is called amino acid binding site. Yes or no? Not, and you can only see this. What is C here? Anticodon loop. How many times I told to all of you? Anticodon loop is what, friends? Where the codons present on the mRNA can come and associate. So this will become D loop and this will become the T loop. That is also very, very straightforward question only. Hence the correct answer will be the B. What did I told you? T loop will help the 3' hydroxyl group to associate it with amino acid. And what did I told you about the D loop? D loop help the anticodon to associate it with the codon. Simple. Hence the correct answer is B. Now, this also surprisingly people has given the wrong answer, but how? Translation means what, friends? Synthesis of protein. In this, there is an important enzyme, amino acyl RNA tRNA synthesis. What is the function of this? Joining the tRNA with the amino acid. This is the function of amino acyl tRNA synthesis, right? Transcription means RNA polymerase. In the DNA, except DNA replication, you can't see the Okazaki fragments everywhere. Anywhere you can't see it. Only in the replication you can see the Okazaki fragments. So A will be the 1, B will be the 3, and C will be the 2. Correct na? It's a very, very, very simple and straightforward answer. Tell me, any one of you, I have not observed this. How many of you answered this question number 30 correctly? Tell me. How many of you answered the question number 30 correctly? Tell me. At least one person, anyone. What you people do if you get a same exam in your JL or DL, what you do? You leave it and come. Okay. Now, see, this question is a purely logic. You know, just if you apply your brain for a half minute, you'll understand it correctly. What is the question says that every student understand me. This is the diagram which is explaining a protein is getting synthesized, right? How many proteins are there? How you can see there is a protein is getting synthesized. Now you have to see how many amino acids are involved in the protein here. 1, 2, 3, 4, 5, 6, 7, 8 amino acids. You can see it. You can see it directly. But what is the question they asked? Which of the following is the first part of the protein molecule that would be translated from the mRNA to? So what every one of you has done, every one of you, this is not the way of thinking. What you have to do, what everyone done, you're all looking about only mRNA 2. Here in the diagram, you're all able to see 8 amino acids. But here, how many codons are there? 1, 2, 3, only 4 codons are there. You tell me, by using 4 codons, how 8 amino acids are involved? No one is analyzing it properly. So you have to see the codons which are present on the mRNA 2, the same codons are where they are present on the mRNA 1. By that, you can easily answer the question. What is the question they says? Which of the following is the first part of the protein molecule that would be translated from mRNA to mRNA to having 4 codons? So which part of the protein is coming here? Okay, why it is not visible? It is visible in the question. 100% it is visible. Okay, so now you only listen. GAA in the mRNA2. Every student understand this is the GAA in the mRNA2. Where it is located? You only see here, it is located on the mRNA here. So this will be the last amino acid, right? So this is what amino acid 8. Here also you can answer the answer. It is a 4th one. Now what is the next codon? CUG is present on the mRNA. Where it is present? 1, 2, 3 here. CUG, which is in which position? 5th position. Then the next amino acid AA5. Next GAC is present. Where the GAC is present in the 1st position. Then obviously it is AAA1. Then CCU, where it is located? CCU located here at the 4th position. Sorry, 3rd position. Then the amino acid 3. AA8, AA5, AA1, AA3. Hence the correct answer is 4th one. What is the question says? The question is saying which of the following first part of the protein they are asking. Suppose this is the protein. They are asking which is the first part. This is they are asking. So you are all people seeing only these 4 codons, but the given number of amino acids are 8. So you have to see not the mRNA, you have to see the codons, where the same codons are present on the mRNA. Then you can interpret it. Okay, it's extremely easy, but no one has answered. I think 1 people. Is it clear now, everyone? Question number 30. Everyone just let me know, is it clear? Question number 30. Quick. Everyone. Okay. It's easy question. Okay. Now listen. Question number 31. It is also very straightforward question, right? Given below are the steps of protein synthesis. Arrange them in correct sequence and select the correct option. You have to select the correct option here. Codon, anti-codon reaction between mRNA and amino acid tRNA complex. No, right? What will be the first step? You only tell me. First step, how many times we studied? tRNA has to be associated with amino acid. So charging of amino acylation or the addition of tRNA with amino acid. So here only you can eliminate these 2 options easily. After the charging, what you have to see here, attachment of mRNA and smaller subunit of ribosome. Of course, that is also a true one. Then later what happened? Codon and anti-codon. So this is the codon present on the mRNA. tRNA contain anti-codon. Then 3, 2, 1. Later what happened, friends? Attachment of larger subunit. That is only we can call the complete initiation complex. Then obviously formation, then to the tRNA. So now which sides got filled? E side, P side, A side. Only P side got filled. Another tRNA comes to the A side. So linking of adjacent amino acid. Finally, friends, formation of the peptide bond. So this will be the correct one. Please mute your mics. Someone is making sound. Please mute your mics. Okay, next one. So this is also question number 32 and question number 33. 70% of the people has given the wrong answer for question number 32 and question number 33. See, I will please mute your mics. Please mute your mics. How many number of times I told you, no one having common sense here. No one behave professionally. How many number of times I told you? You're not understanding what I'm saying. You're all completed PGs and no one knows how to mute your mic also. Look into the question number 32. See, I can take the class through the app itself. There is no problem, but at least other people has to see what is happening, how the questions will be solved. Other people will be benefited. So that is the reason I'm sharing in the YouTube. So please mute your mics. Now question number 32. Repressible operon system is usually found. Okay. Now all of you think and tell me, this is the lac operon. Lac operon is what? Lactose. Right. Now the next one is what? Tryptophan operon. Now you tell me, tryptophan is what, friends? Tryptophan is an amino acid. And what about the lactose? Lactose is the sugar. Now you tell me, what do you mean by the word catabolism and anabolism? If you understand this, you can automatically answer the question. Catabolism is breaking down. Anabolism is what, friends? We are synthesizing something in our body. So what we can synthesize? What we can synthesize? Obviously, everyone remember, we are going to synthesize the amino acid tryptophan. I have already told you, right? Suppose I am a bacteria. I am a bacteria. In my body, I have to synthesize the amino acid continuously. Every single time I have to synthesize it. So it will become what operon? Anabolic operon, because I'm synthesizing. Lactose, if I want to, I am a bacteria, I want to use the lactose. So what should I do? I want to break down the lactose into glucose and galactose. So what I am trying to do here, I am trying to break down the lactose. So lactose will become what operon? Catabolic operon. And I have also told you many times in the operon, friends, lactose is always inducible operon. Is it or not? And tryptophan operon, it is always repressible operon. Is it or not? Inside my body, I am synthesizing tryptophan already. But when you provide me the tryptophan externally, what happens? This tryptophan only will come and block this. That's why tryptophan operon is the repressible operon. Not to get confused with this particular point. Now you only tell me here. Here they said that repressible operon. So which options I can eliminate here? Every one of you. Which options I can eliminate here among these four? Among the four, which options I can eliminate by understanding this line itself? Which options I can eliminate? So easily I can eliminate option 3 also. Even I can eliminate the option 4 also. Is it? Now, friends, there is only two options are there. Repressible operon is anabolic. I told you, right? Here, tryptophan itself acting as a co-repressor, which will go and activate the repressor. Then repressor will block the tryptophan, whatever happening inside the body. So it's a co-repressor. Anyway, inducer is what we studied, lactose, right? So genes for this operon usually switched on. Yes or no? Because inside my body, I am synthesizing tryptophan every time. But whenever the repressor is synthesized, what happened? I have to inactivate it. Hence, the correct answer is option A. Most of the people has not answered this correctly. You have to think it very correctly. Now, just opposite, I have asked another question. So the moment you heard the name of a inducible operon, what you can eliminate now? Option 1 you can eliminate. Option 2 also you can eliminate. Inducible operon system usually occur in the catabolic pathways. Nutrient molecule. Here, what is the nutrient? Nutrient is the lactose. Lactose is acting as what? Lactose is acting as the inducer molecule. If the lactose is not present, what happens to the operon? Operon will be turned off. Correct, na? Then the repressor will become active. Very, 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 very easy it is. Hence, the correct answer is C. Very easy to understand and easy to interpret. But you have to think it correctly. Okay. So in the last NEET exam happened, no? This is the simple question they asked. Lac ZYAI. This is the only question from the operon which appeared in your 2023 NEET question paper. Trust me, when compared to the NEET paper, your JL and DL paper never be tough. That much depth you never ever get. But of course, we have to read it since the syllabus is PG level. This is the same question they have asked in the exam, but they just added I also. Last time I explained you. I is what? Regulatory gene, which is actually going to code for what? Repressor protein. So what are those? LAC-Z for beta galactosidase, Y is permease, A is transacetylase, I is regulatory gene, which code for repressor. Repressor will go and block the operator, not to get confused with that. And this also people has given the wrong answer. Now tell me, every student tell me, what is the answer for the question number 35? Any one of you? And you have to tell me how. First, every one of you answer how many number of ATP we will get. Everyone. See, friends, what is the question they have asked? When the pyruvate is converting into acetyl-CoA. Okay, Mahip already tell me, what is the name of this particular process? What is the name of conversion of a pyruvate into acetyl-CoA? Every student tell me the name of the process. This also has asked a question in your 2023 NEET question paper, which happened on the last Sunday. Direct question. Now, what is the name of that link reaction? That is all I'm asking. No. Morally, Nas, it's not phosphorylation, right? Very good, Mahipal Reddy. It is a decarboxylation. Why you people forgot it? People are saying oxidative phosphorylation. That is completely wrong, right? Okay. Pyruvate decarboxyl. See, friends, it's easy, right? Use your common sense. Pyruvate is how many carbon? 3 carbon. Acetyl-CoA is how many carbon? 2 carbon. So what is happening now? 1 carbon is released out, right? So we are removing the carbon. We are removing carbon from where? We are removing carbon from pyruvate. That's why we can call it pyruvate decarboxylation. So the enzyme involved is what? Pyruvate dehydrogenase is the name of the enzyme. Reaction is pyruvate decarboxylation. And I told you very clearly, it is a reduction reaction where there is a production of one NADH molecule. So one NADH molecule is going to give rise to how many ATP? 3 ATP. Correct, na? Good. Hence the correct answer is 3. As a result of glycolysis, lysis breakdown, glyco sugar. So breakdown of the sugar giving rise to pyruvic acid. The net gain, the question is very crystal clear. They are asking only the net gain and they have not asked the entire cellular respiration. They have asked only the process of glycolysis only. What will be the answer? Obviously 2. Very, very good. Krebs cycle start with the formation of a 6-carbon compound by a reaction between. First of all, what is the 6-carbon containing compound? Friends, every student remember, 6-carbon containing compound only we can call the citric acid. We all know very well how does the citric acid is going to be formed by the combination of oxaloacetic acid and the acetyl-CoA. Yes or no? Because acetyl-CoA is a 2-carbon containing compound. Oxaloacetate is a 4-carbon. 4 plus 2, 6 carbon, giving rise to the citric acid, which is the 6 carbon. Hence, the correct answer is oxaloacetic acid and acetyl coenzyme A. No need to get confused. In glycolysis, high energy reducer compound. I told you, right? NADH2, FADH2, and GTP and ATP. Correct, na? So GTP actually give rise to ATP only. But one GTP give rise to 1 ATP, one FADH2 give rise to 2 ATP, one NADH give rise to 3 ATP. Then obviously the highest, but the question is very clear, reduced. See, if they given the option of FADH2, then you will get confused because both are reduced compounds only, right? But they have asked, they have given only NADH2. Undoubtedly, the answer will be A only. Very simple point it is. Then the role of FAD. 90% of the students has answered this question wrongly. 90% people. Role of FAD. Most of the students are answer that to accept the electrons. Friends, I told you very clearly, FAD, every student use your brain. It is oxidized form. NAD, it is also oxidized form. They never act like a electron acceptors, or they never act like a electron carrier. So now you remember FADH2 and NADH2, both are reducing form. Whichever is in the reducing form, they can act as a electron acceptor and electron carriers. You have to remember that point. And now my simple question to every student, my very, very simple question is, FAD or FADH2, let's consider. What is the name of the, see, in the electron transport chain, we are having 4 complexes, 1, 2, 3, 4. What is the name of the electron acceptor complex that will start accepting the electron from the FAD? I am asking name. What is the name of the second complex? Yes, that is true. What is the name? Very, very good. It is called as what? Succinic dehydrogenase enzyme complex. Correct, na? Where that particular succinic dehydrogenase complex enzyme is present near, I mean to say, which enzyme is involved here? I mean to say, which reaction is involved here? Succinic acid reaction. You remember, succinic acid is converted into malic acid. So here succinic acid is a 4-carbon containing compound only. But this reaction can be catalyzed by a succinic dehydrogenase, which is the second electron acceptor complex. So friends, FAD is going to accept, or it is pump. I can say pump, because ultimate goal, you remember, 1 NADH can pump 10 protons, and 1 FADH2 later, it is converted into how many protons? What we can say that 6 protons. So if FAD wants to pump the proton, first FAD has to be converted to FADH2. So it has to accept the proton. So from where it accepts the proton? It accepts the proton either from NADH or from succinic acid. Then FAD converted to FADH2. Then it is involved in the pumping of 6 protons. Very simple and straightforward question. But most of the people has given the wrong answer. Oxidizer forms never accept electron. They always lose electron. Only the reduced forms only accept the electrons. I want every one of you has to understand that. Answer is C. Which of the following reaction establish a link between the glycolysis and the Krebs cycle? What is the answer for this? Everyone, what is the answer for this? Question number 41. So obviously what it is? Acetylation. See, we read right. What is the link here? Formation of the acetyl CoA. Yes or no? What do you mean by the word acetyl CoA, friends? To the CoA molecule, we are adding the acetyl group. So addition of the acetyl group, we called acetylation. Hence, what will be the answer? Acetylation. Good. The amount of energy released during the high energy bond of ATPs. We have studied now, friends, 7.3 kilocalorie per moles, I told you. But in the given option, there is no 7.3. So which is very near to that? Near to that is 8.9. And some people have chosen 73 kilocalorie. That is wrong. It is 7.3, not 73. And it is not 0.73 also. This much low energy we never get. So it is 7.3. Since they have not given that 7.3 in the exam, so which is near is what? 8.9. That is the answer. In Krebs cycle, we all just now we studied, right? Which of the following enzyme make use of FAD? Direct question. Electron acceptor complex 2, succinic dehydrogenase. Straightforward question. The enzymes common to EMP pathway and C3 cycle. This also most of the students has not given the answer correctly. What is EMP pathway? Embeden, Meyerhoff, and Parnas. Name of the 3 scientists who proposed the glycolysis. So EMP pathway or the glycolysis is same. And C3 cycle. Now you only tell me, What are the two enzymes are involved? In which is common? Aldolase. My friends, you remember what is the function of aldolase? Which breakdown? Which is involved in the breakdown. And next one, triose phosphate isomerase. All the students has to ask me, sir, how they are similar. That is also you have to ask. Let's talk about the triose phosphate isomerase enzyme. In the glycolysis, what do we get? Everyone remember, we get DHAP, we get G3P. Yes or no? These two can be isomer, right? Both can be interconvertible to each other. And friends, you all people remember or not? In case of a C3 cycle, we get glyceraldehyde 3-phosphate and another one is what? 3-PCA, phosphoglycerate. These two also isomer, right? Here also isomerase enzyme only. Here also isomerase enzyme only. Yes or no? Correct. Then aldolase. You remember, friends, in case of a aldolase, it is breaking down fructose 1,6-bisphosphate in the glycolysis, break down into DHAP and G3P. What is the name of the enzyme? Fructose 1,6-bisphosphate aldolase enzyme only it is. The same enzyme is involved in the C3 cycle also. In breaking down of what? 6 molecule of GA3P. Out of this, 1 molecule of GA3P come out and 5 molecules will be regenerated. And you remember, any one of you remember, this 1 molecule of GA3P, what happens to it? It produces glucose. That is also correct. Now you have to ask me, sir, then where we use aldolase? Friends, you remember this 5 molecule of GA3P, they are giving rise to what once again? They are giving rise to 3 molecules of RUBP. So during this regeneration process, we use aldolase. Whenever we are converting this particular 5 molecule of GA3P into 3 molecule of RUBP, we use the aldolase enzyme during the phase called regeneration. This also many people has given the wrong answer. In the NEET exam, it was really, it was really shocking. Every time they used to give only 5 questions or 6 questions in the NEET from the plant physiology. This time they have given 13 questions only from the plant physiology chapter. So you people, whomever reading the botany, you try to understand the importance of the plant physiology in your entire syllabus. And cell biology, including the biomolecules, they have given nearly 10 to 12 questions. Then you people has to give the highest importance to the cell biology and this also. Molecular basis of inheritance, they got 6 to 7 questions. Genetics, they got 7 to 8 questions. No, opposite. Molecular basis, they got 7 to 8. Genetics, they got 6 to 7. So you there, and Monera, Protista, Plantae, the entire what we can say, Bryophyta, Pteridophyta, Gymnosperm, Angiosperm, all together they got 10 questions. So, but for you people, it is entirely different. Then imagine how many number of questions that you can expect. So because your question paper never be that much tough when compared to them. So you should always think it correctly and you have to, focus and give the importance to the chapters. Okay. Now, this also, when I'm explaining you the lesson, I have explained you difference between photophosphorylation, oxidative phosphorylation. Anyway, this option A is wrong. See, friends, in both phosphorylations, we produce ATP only, but that is not the question. In a 4 options, they have not talked anything about ATP. Here only they talk ATP, but in other they talk ADP. So this option is irrelevant. So you have to remember, they're asking with respect to the electron acceptors, friends. In oxidative phosphorylation, what happened, friends? You remember at the last, what happened? Oxygen combined with 2 protons, oxygen combined with 2 electrons, giving rise to the water. So in the oxidative phosphorylation, we use oxygen. Photophosphorylation, what happened? Every student remember, right? When water molecule break down, same opposite reaction. I hope every student is getting me. 2 protons, 2 electrons, and half oxygen will be released. This is the oxygen we breathe. So my friends, In the photophosphorylation, oxygen will be released out. In the oxidative phosphorylation, oxygen is using. You can see, right? So both are opposite. So that is the answer, option B. In which step CO2 is not released? Surprisingly, this also people has given the wrong answer. You only tell me, friends. Glycolysis, how many steps are there? 10 steps. You only think in any 10 step, do we release any carbon dioxide? No. In the glycolysis, my friends, we only release a water molecule. Please remember my words. In the glycolysis, we have never released anyone. Lactic acid is what? You tell me. Pyruvate. Pyruvate is how many carbon? 3 carbon. Lactic acid is how many carbon? 3 carbon. So where there is a release of carbon dioxide, here also carbon dioxide is not released. Malic acid into oxaloacetic acid. Malic acid is 4 carbon. Oxaloacetic acid also 4 carbon. So where there is a release of carbon dioxide, here also carbon dioxide not released. Hence, what will be the correct answer? All of this. In any of these, no release of carbon dioxide. Simple point. Which one of the following is responsible for transfer of electrons? It's a direct question. You people has to remember this. Involve transfer of electrons. Iron sulfur protein, it is not involved in the transfer of electron. Phytochrome, no. You know, right? A phytochrome will be involved in the absorption of light. You know, right? F0 and F1 is involved in the ATP synthesis, which we can call ATP synthase enzyme. Of course, they have not given F1 also. Cytochrome is the only one, because we read, right? Cytochrome A, cytochrome A2, A3, cytochrome B6F. They are the one what involved in the electron transport or transfer of electron. Hence, the correct answer is cytochrome. The number of glucose molecule required to produce 30. This also people has given wrong, you know. Under anaerobic condition by yeast cell. See, friends, under the anaerobic condition, every student know very well how many number of ATP you get in the anaerobic condition. You get only 2 ATP, you will get it. For 1 glucose, under the anaerobic condition, you get only 2 molecules of ATP. But the question asked is how many molecules of ATP are required? Sorry, how many number of glucose are required to produce 30 ATP? 38. 1 glucose 2 means you require 19 glucoses. Every student remember my words. Anaerobic respiration, we get 36 to 38. Anaerobic respiration, we get only 2 molecules of ATP. In the aerobic respiration, we get 36 to 38 molecules of ATP. Always remember. Now they're asking only anaerobic respiration. So answer is 19. The inner membrane of mitochondria is very, this also, almost 10 to 15 people has answered this question also wrong. The inner membrane of mitochondria is very selective in respect to entering molecule. A molecule that regularly enter in it is, some students has given answer pyruvic acid, right? But why? That is my question. Because, friends, on the inner membrane of mitochondria, we have a shuttle systems. Correct, na? So that shuttle systems can easily allow the pyruvic acid to enter. Glucose never enter. Glucose will enter. This also asked a neat question in the last week. Glucose will follow the facilitated diffusion sometime. Sometime it will show the active transport. Anyway, ATP, because it's a phosphate group, never travels. Oxaloacetic acid can travel, but not directly. So directly which can enter is undoubtedly pyruvic acid. This is the right answer. Question number 50. What is active glucose? See, in the glycolysis, the first step, you tell me what happened. Once the glucose enter into the glycolysis, what is the first step? Glucose immediately convert into glucose 6-phosphate. Why, friends? If the glucose convert into glucose 6-phosphate, phosphate is what charged? Negatively charged. Negatively charged. Do you think it crossed the plasma membrane? Never. So it can't escape back. It can't escape back. It has to only continue the remaining 9 steps. So friends, and glucose 6-phosphate. Phosphate is what? Energy, right? So active form of the glucose is obviously phosphoglucose, right? So some people will think that glycerophosphate. Glycerophosphate is not a direct form of glucose, is it or not? Glucose here 6 carbon. When I break down the glucose, I'll get the glycerophosphate, which is only 3 carbon. So this cannot be an active form. So phosphoglucose is the most active form, right? Is it clear, everyone? Now you can ask me any doubts in the 50 questions. The question paper is easy, but some of the questions are very logical. The people who score a 65, that is really, really good. But I want every one of you focus very much. Okay. Now you can ask me any doubts in these 50 questions. I will re-explain to all of you. Now, for the next week syllabus, I am telling you right now, because from tomorrow, iron, sulfur, proteins, they are the part of electron acceptor complex 1, and they are the part of electron complex 2. So iron and sulfur, they can act like a cofactors. They are not act like a electron carriers. Okay, they will only act like a cofactors. Okay, so tomorrow, because in the biochemistry still we are left. What we are left in the biochemistry, I'm telling you the syllabus for the next Sunday. Please don't ask me again. Now I want you to remember right now only. Okay, if you want, write it. So we are left with enzyme kinetics. By tomorrow morning class, enzyme kinetics will be over. Freshly, I will teach you enzyme kinetics. Then lipid chemistry and lipid metabolism. So only these 3 topics are left in the biochemistry. These 3 you have to, so these 3 from the biochemistry. Molecular biology is done. We have DNA repair. DNA repair we always read in the genetics chapter. Now, these 2 from the biochemistry, and remember everyone, entire rDNA technology, entire. Okay, only rDNA technology, that's all. Not the tissue culture and all, because we have finished only rDNA technology. This is the test for the next. From rDNA technology, I will give 30 questions I will give. From enzymes and kinetics, I will give 10 to 11 questions. From lipid chemistry and metabolism, I will give 9 to 10 questions. So entire rDNA technology, enzyme kinetics and enzymes and its kinetics, lipid chemistry and lipid metabolism. Okay. Clear, everyone? We'll continue tomorrow morning 5:10 a.m. and we will continue the kinetics. Within 2 days, it will be finished. Just now I told you, right, Varalakshmi, NADH, NADPH, NADH2. Okay. These 3 are reduced forms. Okay, 3 are reduced form. Varalakshmi, remember in a simple way, wherever you can't see the hydrogen, remember that it is the oxidized form. So NAD is the oxidized form. These 3 are the reduced form. 3 are act like a electron acceptors and electron carriers. NAD only lose the electron. Got it? Okay, perfect. Please revise, and the people who scored very well, like people who scored above 68 to 84 or 85, don't think that this is my best performance. Okay, we should never ever think that this is my best. You should always think that I have to concentrate on every single test. I don't consider your great performance in 1 or 2 tests. I will always see the performance of an average number of tests, because I will give you one example right now only. The people who scored very well in the first test, most of the people scored very less. The people who not scored well in the first test, they scored very well in the second test. So 1 or 2 tests never decide anything. So the people who scored very less, no need to worry about it. Always take it as a great challenge and read every single point. And 1 second, just listen. For the lipid chemistry and lipid metabolism, I'll show you. I have already shown you, but Once again, I will show you what kind of study material that I want you people to read. Okay, so please read that. Please read. If you don't read, there is no point of explaining to you. Okay, because if I only explain, you never succeed. You have to read that correctly. rDNA technology also. rDNA technology, I will give you one PPT, one PDF, and keep the PPT with you and read it correctly. When you are reading the rDNA technology, I want every student has to read compulsory, mandatorily name of the scientist, mandatorily. So now remember everyone, not to get confused. Whenever you are reading the enzyme and its kinetics, all you have to go to cell biology. In the cell biology, there is a biomolecules PDF. Read in the biomolecules PDF, you will get the lipid chemistry also, enzymes chemistry also. You can't get the lipid metabolism. You can't get it because that is the PG syllabus. I read the PPT for that. So lipid metabolism, read the PPT. Lipid chemistry, enzyme and its kinetics, read this biomolecules. Not to get confused with that. And you see here, rDNA technology, I have kept one PDF here under the chapter called as rDNA technology and genetic engineering. This is purely PG level. I will provide you the another UG level also. Read both, or if you can't read the PG level, at least read the UG level along with the PPT. That is sufficient. Okay, perfect. Keep revising. Don't think that we have achieved everything. I want every one of you to read very patiently. Yes, Srinivas. Biochemistry syllabus is very large for the zoology people when compared to the botany. Botany, they are having nothing. Botany people not having amino acid also. We have to read lipids, amino acid, carbohydrate chemistry, everything. Okay, it's there. It's there. If it's not there, I never tell. Okay, perfect then. I will see you in the tomorrow morning class. Please revise it once again without fail. I will share the winners list and cash prize by evening or in the night. Okay, thank you all. Thank you.